Good morning. Thanks, Mark, and thanks to the broader AIS team for inviting us to speak today. My name's Ryan Beal. I'm the CEO of Dyve Biosciences. Dyve is an R&D focused company that's broadly enabling transdermal drug delivery. We've developed this technology to be be able to treat multiple disease states with multiple active ingredients. We've got human proof of concept data spanning a number of indications across a number of drugs, as diverse as adipolysis and alopecia, psoriasis, melasma, and gout. We have advanced clinical programs in gout and melasma, both expected to enter phase 2 trials next year. I'm going to spend some time talking about our melasma trial, but first, the mechanism of action. Dyve works by essentially opening up two important transdermal doors: the stratum corneum and the tight junctions. We work by fluidizing the stratum corneum and then modulating the tight junctions, in essence to open up physiologic valves, allowing us to create pores or channels through the skin and deliver, much like a needle would, deep into the bloodstream. Dyve 600 is a good example of this technology. In this case, we're packaging tranexamic acid with our delivery system to take the drug deep down to the melanocytes. Why were we interested in melasma? Well, in addition to the broader market dynamics, high unmet need, big prevalence, the real reason we got interested was because the drug candidate. It's a proven drug with an established safety profile and some, at least, hint of efficacy when delivered orally or by injection. Tranexamic acid has demonstrated some modest efficacy in melasma when it's used for other indications. And there was an interesting study out of Korea where they did a pilot study with microdosing tranexamic acid for melasma and saw decent results. So we said, given what they've seen with microdosing, given the safe profile, is this an ideal candidate to improve the efficacy with our transdermal delivery system? And that's the model we've used for a number of different products. Melasma, as you know, is a disease that starts deep at the melanocytes. It's beyond most topicals' ability to penetrate. That melanocyte is essentially a volcano creating melanosomes or lava that rises to the surface, resulting in the discoloration or pigmentation we see at the surface. The problem is getting deep down into that melanocyte. So our question was, can we open up some of those valves and deliver the tranexamic acid levels? Not only were we The quick and short story is yes, when you look at blood serum concentration levels, not only were we able to get tranexamic acid into the body and into the blood, but we exceeded on a dose equivalent basis what you'd see with orally delivered drug. While this may be challenging for some to believe, the rationale is quite simple. If you can bypass the acidity of the gut and you can bypass first pass metabolism, you end up with a very efficient delivery system. And that's what we've done here with tranexamic acid, as well as a number of other drugs. Outside of the blood serum studies, we've got a robust clinical program underway, of course, starting with our feasibility studies, but also doing a number of split face tests. And ultimately, right now, we have a full face study ongoing, comparing various iterations of our formulation. This is an example patient from our initial feasibility study, Dyve 601. This is one of Mitch Goldman's patients down in San Diego. She was a treatment-resistant melasma patient that really at baseline was being maintained on a course of on and off of hydroquinone, various laser treatments and peels, just to stay where she was on the left. After layering on a topical with our delivery system for 4 weeks, you can see a rapid resolution of the melasma, showing that when you can squelch that melanogenesis at its source, you really can have profound effects. What's interesting is in split face studies, which we've done a number of, when you treat half of the face, in this case, this was a split face study where half the face received active, the other half received control. Due to the bioavailability, you end up getting results, equivalent results on both sides. Here are some example patients from our Dyve 603 study. This is the control side. And you see nice resolution of some of the melasma and an overall improvement in skin tone and quality on the side that didn't receive drug. Of course, ultimately, as we build out our clinical plan and our commercial plan, we don't intend on treating half of the patient's face. We want to treat the full face, and we want to optimize the technology to really increase the residence time of tranexamic acid in the target site down by the melanocytes. This is an interim data from our ongoing study where we're working to modulate that technology, opening up that top valve at the stratum corneum level, but titrating down the lower valve and the tight junctions in an effort to increase the residence time of the drug at the site of action. This study just completed enrollment. We'll have final patient out later this summer and a full data readout later this year. So the question I always get asked is, well, what else does this work in? What else can you do? What excites us most about the technology is its broad applicability. What we've learned in aesthetics and modulating the skin for penetration has led us down a very interesting path on the therapeutic side. And in particular, we have some tantalizing data on oncology, where we're delivering alkalizing and buffering agents to the acidic cancer environment, the tumor microenvironment of the cancer, and showing some incredible improvements in survival rates by decreasing the metastases. So we're excited about the potential of transdermal drug delivery and what we can do with our technology. We think we're ultimately going to be able to unleash the true potential of what can be achieved with transdermal delivery. Thank you.